Hello, everyone, and welcome to the Immunotherapy Treatment Chapter, brought to you by the Myeloma Crowd Community Program by HealthTree. My name is Audrey Burton-Bethke, and I am the Myeloma Crowd Community Manager. We are very excited for today's event, and I will begin with some intro announcements before we officially get started. Today's meeting will be recorded, and the recording will be sent out to all registrants within 48 hours of the event's conclusion. As always, I'd like to thank our sponsors, without whom this would not be possible: Bristol Myers Squibb, Takeda Oncology, Karyopharm Therapeutics, Adaptive Biotechnologies, Oncopeptides, Janssen Oncology, AbbVie, Sanofi, Amgen Oncology, and Genentech. The event agenda for today is as follows. My introductions will last just a few more moments, and then I'll turn the time over to the president of the HealthTree Foundation, Jenny Ahlstrom. She will present our two highly anticipated featured guests today and then moderate the rest of the session. As you know, the event for today will consist of questions that you, the audience, have submitted previously. Doctors Allison and Sharma will answer these questions to the best of their ability. We feel very privileged to have them here with us today, and we thank you both for sharing part of your busy schedules with us. Dr. Allison and Dr. Sharma, the audience here today has had the privilege of watching the Jim Allison Breakthrough documentary. Because of this experience, we have received many emails and messages praising the persistence and innovation that brought the advancements of immunotherapy through your hard work, and we are so grateful. We are looking forward to this session, and thank you again for being with us today. It is now my pleasure to introduce Jenny to you. She is a myeloma survivor, patient advocate, wife, and mom of 6. She is a believer that patients can help accelerate a cure by weighing in and participating in clinical trials. She is the founder of the HealthTree Foundation, which consists of the Myeloma Crowd by HealthTree organization and the HealthTree community for AML. Jenny, the time is now yours.
Thank you so much, Audrey. And when Audrey told me that we were going to have you both on this in person, I was just totally blown away. And I will try not to cry during today's session, but I'm just so amazed by what you've done, and look forward to talking to you today. So let me just give a small bio for both of you before we get started. Dr. James Allison is Regental Professor and Chair of the Department of Immunology, Olga Keith Weiss Distinguished University Chair for Cancer Research, Director of the Parker Institute for Cancer Research, and the Executive Director of the Immunotherapy Platform at MD Anderson Cancer Center. He spent a distinguished career studying the regulation of T-cell responses and developing strategies for cancer immunotherapy. He earned the 2018 Nobel Prize in Physiology or Medicine, which he shared with Dr. Suku Honjo for their discovery of cancer therapy by inhibition of negative immune regulation. His current work seeks to improve immune checkpoint blockade therapies currently used by our clinicians and identify new targets to unleash the immune system in order to eradicate cancer. Dr. Sharma, Dr. Padmanee Sharma is, you go by Pam, right, sometimes? Is professor of genitourinary medical oncology and immunology in the Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center. She's participated in 54 research projects since 1996, focusing primarily on immunotherapy in her collaborations with Dr. James Allison. Dr. Sharma is a Cancer Research Institute investigator who's tested new prostate cancer immunotherapies, looking for differences in T-cell subsets and function between pre-therapy blood and tissue samples versus post-therapy blood and tissue samples. She has won multiple awards during her career, including the 2012 MD Anderson Cancer Center Faculty Scholar Award and the 2008 Melanoma Research Alliance Young Investigator Award. With Dr. Allison, Dr. Sharma is exploring combinations of immunological therapies and targeted drugs in preclinical studies to more effectively treat a variety of cancers. So thank you again for joining us today. It's just an honor of a lifetime to meet with both of you. As an MD Anderson patient myself, I could have really never imagined that I have the privilege of interviewing you two together. I know you have all watched the documentary, so you know that you are married, and your story is truly, truly stunning. I also saw you speak at the Stand Up to Cancer Summit as a patient advocate prior to COVID, and that was a delight to do that. Now on the call today, we have mostly patients with blood cancers that include myeloma and AML. They have precursor conditions, might be newly diagnosed, or might be 20-year-old veterans. But we'll be talking about immunotherapy in general today as well, and we'll try to get as many questions as we can today in the hour that we have. I really want to take a broad look first and think about just innovation in general. So as an entrepreneurial family, we kind of treated my myeloma as a startup. And there's a clear difference between incremental innovation and disruptive innovation. And your sheer will and just way of seeing the world differently and unconventionally made ipilimumab happen. And that launched a whole new immunotherapy treatment paradigm. So you're an innovator and a leader. And in the tech world, knowing how to replicate this success means knowing why you succeeded in the first place. And a lot of entrepreneurs actually don't know why they made it big. So I'd like to kind of ask, what were the key factors of your success and what's replicable in that success? And I would love for both of you to answer these questions as we go through our session today.
Well, if I could start, I think that the key to success was that I really wanted to understand the mechanisms. I'm a scientist, I did not get into this to try to discover directly say, oh, I'm going to find a cure for cancer. I said, I want to understand how the immune system works. And every time I figure out a step, every time I describe and understand a particular step that regulates immune responses, I will think about how we might be able to use that or not, and then move on, and it was basic science. It was data. And finally, when I had the idea that taking these negative checkpoints off, blocking them, and letting T cells, just do their thing, without the inhibitory pathways that are normally there for a time. You can't let it go forever, but would eliminate cancer. We understood the molecules, we understood the details of the mechanism after 15 years of work, and then it was time to say, let's try this. And but anybody could tell me why they thought it wouldn't work, and I was armed. I knew more about it than anyone, and could just say, well, if you say this will never work, it's because you're ignorant and you're not willing to take a chance. So look at the data. If you find a problem with the data, I'll talk to you. If you're just going to say it'll never work, then I'm not interested in talking to you anymore.
Yeah, I have to agree. I think it's all about following the science. I mean, if you stay true to the science and you look at what the data is showing you and you follow that up with experiments to test hypotheses to make sure that you're looking at your data in multiple different ways and understanding what the data is saying to you about that specific mechanism, then you can move forward in thinking of how to translate that into the clinic. So I think it really starts from the basic science and making sure you have that understanding.
After the basic science and that this will or won't work, there was such a process to get from start to finish. What else was successful in that process?
I think after a time, it took a while, but convincing the clinicians who were used to thinking of cancer drugs as drugs that killed cancer cells, when the drug that I developed, the approach had nothing to do with the cancer cell. It unleashed the immune system. And so to treat it like a cytotoxic, like, it's something's going to go and just immediately kill cancer cells and you're going to get a response. I had that was the second big hurdle when it went into the clinic, is convincing them not to treat it as just another cancer drug, but to realize the biology is different and you have to accept that. And that was touch and go for a while, but finally they came around.
And then all the work with, go ahead, Dr. Sharma.
No, I was just going to agree with that. And I think, one of the big things is to find collaborators who, can look at the science with you and help to move the field forward, right? We had so many failures in the clinic with people trying to target the immune response. There were so many failures. And so I think, although you do have failures, there is a way to go back and make sure that you look at the science carefully enough to say that you're not going to throw away the baby with the bathwater, right? So that, there are certain things that maybe we targeted the immune system in ways that didn't work, but that did not mean that we throw out the entire concept the immune system being a powerful way in which it can specifically target cancer.
I think that's a lesson in any entrepreneurship endeavor is don't be afraid to fail. Failure is okay.
Yes.
And sometimes in healthcare, it's a challenge. Dr. Allison, you got a comment?
Well, I was just going to say, it's an interesting point that you make. Don't be afraid to fail. When I was trying to decide my career goals, my dad was a doctor, and I realized that in medical training, you have to get your head full of facts, and the person presents and you develop an algorithm and say, this is how I'm going to treat you. And you better be right, but the reason I drifted towards science was I'm interested in being on the edge. I want to move knowledge forward. And so you're going to be wrong a lot if you do that. And you should celebrate being wrong. Not to be wrong intentionally, but just say, okay, I learned from my failure and I'm going to move on and not make that mistake again, but all you have to do is be right, occasionally in a big way, and it pays off. If you don't do that, all you're going to do is incremental.
That's what I want to talk about next. It seems like in science, it seems like the system is not necessarily set up to cure diseases, because a lot of times there is like a lot of pushback, and there's a lot of, you get what you measure sometimes. And so a lot of researchers are rewarded with grant funding for publishing, and maybe they should be given cash prizes for advancements or cures instead. So, I could see how we could systematically go after a disease and give the inventor of the cure what we would have spent on that disease for a year in Medicaid or Medicare, So what needs to change in order to be able to replicate your type of success in the system as a whole?
I don't know if there's any simple answer except just to stress that science is funded now in 3-year cycles, for example, and so you have to have something in that almost positive finding. And usually the only way you're going to do that is if you're following everybody else and doing the same thing everybody else is doing. You move along a little bit. But if there's something really revolutionary, then I think that there has to be the courage to put the money there. High risk, high yield. They're different agencies now beginning to do that more. They set aside a certain amount of money. Some money on the bulk of the money on the sure bets. Where you're just going to continue to make little tiny steps, but it's all in the right direction, as opposed to some money where you say this may be wrong, but if it works, we're going to be light year ahead.
Yeah, well, that's how you looked at it, and you leapfrogged. You did more work that could have been done in 20, 25, 30. So in my opinion, it would behoove us to encourage more people like you to make the process easier for you to come up with those major advances. And so I know, like, maybe earlier FDA approvals, or like on phase 2, because you've got some safety data already, and you've got some efficacy data, or I'm not sure what else, but
we have to make sure that innovative science is really where we invest a lot of money into it, right? First, innovative science has to be a priority. It can't just be an afterthought. So instead of making the incremental science the big payoff, it should be the innovative science, the disruptive science. We should be looking for those projects. Secondly, we have to fund junior investigators at much larger intervals, instead of just saying to them within 3 years or 5 years, tell us something new. We have to fund them for longer periods of time. And there are some organizations that do that and do that very well, but not, I don't think, enough, right? We need the best and the brightest minds to come into science. And if you want that, then you have to make science, an endeavor that people are not questioning where's their next salary or paycheck coming from, how are they going to support their families? Because if you think about it, when you come into the scientific arena, your paycheck for a postdoctoral fellow can be somewhere around, $50,000 a year or something like that. Is that enough to support somebody and their family? Do we need to do better? Because if you want to grow the best and the brightest, you have to attract them in. It's why Wall Street's done so well, right? They don't pay those kinds of salaries that we're paying in science. So how do we change that? And how do we make sure that the scientific projects are funded well enough so that they can continue to stay there for a long period of time? until we can see these big advances that come forward. And then the other thing is the clinical research dollars. A lot of times we're paying lots of money into clinical research for a clinical endpoint. Did they have an overall survival benefit? Did they get their tumor shrinking? But we're not putting enough research dollars into the clinical trial to say, did you get a biopsy sample to understand how the drug is really impacting the human immune response? Did you actually look at the tumor tissues themselves to see what the changes were so that we can understand why the drug worked or why it didn't work? So what do we need to do differently? So those are things that I think need to be addressed better.
Yeah, you can learn just as much from a failure as you can from a success in a lot of ways. And so, but there's no premium on that right now. There are 3,000 trials of chemotherapy, of immunotherapeutic agents, just looking for a clinical signal without any biopsies, looking and saying, well, what happened? Did you almost get there? You're looking for a home run, but did you get a double? And the only way you know that is if you spend the time to get the data, and every patient should count. Every patient should be studied, not just for a clinical endpoint, but for what actually happened in that patient. So you don't need 200 patients to get an answer. You need 20.
And why doesn't that happen now?
I think, again, it's a funding issue, right? I mean, the pharma companies really want the home run because that's where they're going to get their big payout, FDA approval. It's easier to say, okay, the drug didn't give me the clinical endpoint, so I'm just going to move on to the next drug in my pipeline, rather than to say, okay, I'm going to spend a lot of time and money into understanding why this drug didn't work. I think it's just who is involved in making the decisions. So if we as scientists want to make different decisions, or patient advocates want to make different decisions, then we have to make sure that the funding is in place to get a biopsy. Every time we want to biopsy a patient, it's $5,000. We're not going to bill this to insurance, they're research questions. So we have to come up for how to pay for those things.
Part of that's because of this new era of immunotherapies, because before, cancer was about killing cancer cells, and that was it. You either killed them or you didn't. And the immune system be damned. Nobody cared. Nobody thought the immune system. But now we know the immune system is very complicated. There are myeloid cells. They're called, that can suppress Tcells, the one that do the damage, and you've got to get them all working in concert. And we're learning a lot about how that works at the molecular level. But if you don't look, you're not gonna say, okay, well, I didn't get 100% there, but I got 30% there. So if I add this 30% and that 30%, now I'm 60%. If I add another 20, then I'm 80, and that's the way we have to look at it. Not just one drug and say, I'm going to kill all the cancer cells. That doesn't work.
Yeah, that's so fascinating, and that could be related to prior lines of therapy too, right? So if patients have been on another prior immunotherapy and then they get another one, like what's happening to their immune system along that path? And is that better or worse? Yeah. And Dr. Sharma, you had a paper written about like standardization of labs and things like that too. Do you think some of those types of things would help advance the science faster?
I think if we share how we're doing translational research, the standard operating procedures for how samples are collected, how are they transported to the lab, how are they processed, how are we looking at human immune responses from these samples? Yes, I think those things will help. I don't want to say standardization because we all need to use the same technologies, because I think we have to be innovative in technologies as well, so that we have to, if one lab developed a whole new way of looking at, myeloid cells, then we should all learn from that. And so I don't want to take away from innovation, but I do want to make sure that we understand how each of us are going about using it. And if there are ways that have worked for lab A, then lab B through Z should use that for getting the samples and moving it into the laboratory. Maybe we all need to say within an hour, we were able to get the samples out of the operating room or out of the pathology suite into the laboratory. That makes a difference because cells are, alive, and we're able to do the assays that we want to do. So there are things we can do to optimize and make the process better.
Okay, great. And a follow-up question, Dr. Allison, or both of you, how do you teach your students to innovate? I mean, you had this personal mission, right? You were a force of nature. So to me, you can say persistence, but it's way beyond persistence. It's just you're willing it over the finish line. And this deep sense of curiosity. And then what can patient advocate groups, well, that's a separate question, but how do you teach innovation to students?
Well, what I try to tell my students is when, it's sort of like a band in a way, but you got different people with different expertise doing different things. And periodically they present their findings to the group and say what they think it means. And when I challenge everybody in the room, I say, I want you to give me another explanation that's equally as plausible as that one that you just heard, that it would fit the data equally as well. And that's how you move it forward. You don't just say, oh, obviously that's right. And you stop and say, you figured it out and say, no, question. Is there another explanation? How do you know it's, in the case of this work that we did with CTLA-4, people assumed it was a positively regulated molecule. We said, no, what if it's a negative? I mean, that's black and white. Nobody was asking whether it was a negative because nobody had ever found a negative regulator before. So, you just say, oh, but giving a positive is the same as taking away a negative. So what's the experiment you were designed to know the difference, So that sort of thing.
Yeah, challenging them to think differently and pushing them.
Absolutely. We always encourage students to read the literature, but also to make sure that you're critical, right? You're not just accepting everything that you read. Be thoughtful, look at the paper and say, is this how you would have done it? Is there a different way that you would have done it or thought about it? And even do that with your own experiments. And in lab meetings, definitely do that with each other's lab presentations. So yeah, I mean, be disruptors, disruptive thinking is what we hope everyone's participating in when they're, within the lab environment.
Mm-hmm.
Yeah, it's a subtle thing because you can't just say attack, prove somebody wrong, but you can prove they're wrong in a nice way.
Mm-hmm.
Wait a minute, have you thought of this?
Yeah.
And then they can go on and they can, you're helping them, oh, I didn't think of that. Now I'll think of that and I'll go do it,
Yeah. Asking questions,
Yeah.
right? Instead of criticizing is a good way to think about it. Okay, let's talk about immunotherapy in general. Thanks to you, immunotherapy development is growing in oncology, and we have checkpoint inhibitors, monoclonal, bispecific, trispecific antibodies, antibody drug conjugates, T-cell therapy, NK cell therapy, vaccines. I'm probably missing more. I once asked a myeloma specialist what would be the optimal cure in myeloma, and he said to have your immune system recognize the myeloma and kill it. And I thought that was a pretty great answer. So how do you identify, like you did, where the immune system has gone wrong for any kind of cancer? Maybe you already answered this because it's kind of like the basic science you were talking about earlier. But do you have any other thoughts on that question?
Well, I guess what we found was that you can turn the immune system off by recognizing stuff in the wrong context, the wrong way, was basically it. And so it's not all about getting it turned on. It's making sure that it's done in a way that's productive and not counterproductive. So I think that first you got to find what can be recognized on the myeloma cell, and we're both T-cell centric, because T cells have the ability to detect a 1 amino acid change in a protein that may be caused by a mutation associated with cancer. And so that ultimately, I think, can be the goal, although it's easier stated than done. And but I think where we're headed is we understand more about the process as a way of genetically identifying changes and specifically trying to retarget T cells to go after them, probably on an individual basis.
Yeah, we're trying to learn about each microenvironment. I mean, I think the foundational work that Jim's lab did was about how is the immune system regulated? How are T cells regulated? Just to understand T cells by themselves, how do they regulate their function? And then once he understood that, then how do you take advantage of that knowledge to use it in cancer? So I think now we know, though, that it's not just T cells, right? They're dendritic cells, they're myeloid cells, they're NK cells. So again, from a basic science standpoint, how is each cell regulated? How's a dendritic cell regulated? How's a myeloid cell regulated? So we have to go back through that basic science part and then understand how to combine what we learned about T cells with each of those other cell subsets. So that's one way to move forward. The other thing to think about is that which microenvironment takes advantage of which particular immune evasion tactic.
Yeah.
So is it in prostate cancer that the immune evasion is no T cells in there? So now we have to figure out how to bring T cells in. Is it in glioblastoma that we have lots of myeloid cells? So we have to figure out how to target the myeloid cells. And so in each microenvironment, we may need to come up with a personalized kind of strategy for that particular treatment in order to have success.
One of the things that we're realizing too, particularly along the lines of what Pam was talking about, in myeloid cells in particular, but I think to some extent in T cells as well, is that they're shapeshifters in a way. They adapt to what their environment is like, and they try to deal with it in a way, and they'll change. You know, you come in with your drug and you change something, and the environment changes. You know, the environment responds to the perturbation that you did, and it can either help you or hurt you, and you need to know which way. You know the details. The cells can. You know, we're used to, as people, you put stuff in boxes, you know, so this is this, this is this, this is a lot of boxes, but the boxes have very soft walls. They're very porous walls, turns out. And what's in one box can pop up in another box, you know, in a matter, you know, very quickly. And we're just beginning to learn about how that occurs.
Well, there's so much interaction between it. So a question is CAR T cell research, which is now getting to be pretty big in myeloma, and I know some in AML as well, and other blood cancers, a lot in leukemia, lymphoma. Is that a precursor? Or is that after the fact, after your immunotherapy discovery, and kind of a follow-on of that?
So a CAR T cells really, again, is taking advantage of what we know about T cells, right? So T cells require a T cell receptor that can recognize an antigen in the context of MHC. So what the CAR T cells did is that they just created an antibody that could recognize the MHC in that antigen, basically, and put that onto the T cell so that it gives it direct signaling into the T cell as if it's the T cell receptor recognizing that. So it's an engineering technique to take advantage of specific antigens. But CAR T cells at this point are limited by what the specific antigens are. So we need to know what those antigens are in order to generate an appropriate CAR T cell. I think more and more people are trying to understand how they can take advantage of even more engineering capabilities so that maybe we don't need to know every specific antigen, because let's say you identify antigen X and the CAR T cell eliminates all the tumor cells with antigen X, but then the tumor cells with antigen Y just keeps growing, right? And that could be a problem. So how do we take advantage of, you know, getting the T cell to respond and then, you know, allowing epitope spreading, for example, to occur so that you can get tumor cells that have antigen X and tumor cells that have antigen Y all eliminated at the same time. So what are the combination strategies, or what are the engineering strategies that will get us there? So I think, yeah, we're at the tip of the iceberg in understanding these things, but certainly there's a lot of research being done to improve on what we currently have.
Okay, great. Dr. Allison, did you have any comments?
Yeah, I guess what I would say is the fundamental difference. I wouldn't say necessarily that CAR T cells came out of our work. What I wanted to do was come up with a way where we could unleash the T cells in your body by pressing on the gas pedal and taking off the brakes and stuff like this to get the ones that are responding naturally. CAR T cells are genetically making that happen and giving, you know, 10 billion cells all at once into somebody instead of, you know, growing them up. Because in our case, we don't even need to know what the antigen is necessarily. To get it to work. But the CAR T cells, you have to know exactly what that target is, and also know that it's not on normal cells, because you'll wipe them out too, and that could be catastrophic. You know,
In some cases, not in B cell malignancies.
Yeah, in B cell malignancies, it doesn't really matter. You can take out all the B cells, but if turns out that the thing you're targeting is on heart muscle, you know, and just, you know, and for some reason that you don't understand, then you know, you got a problem. They work spectacularly well in B cells because there are numerous targets that are just on those B cells and nowhere else in the body. So,
and as these therapies are being rolled out in blood cancers or solid tumors, is there a way? I've heard some experts talk about, well, we need to make sure that your immune system is robust enough to take advantage of some of these things. Like, is there going to be, or is there anybody working on, what does that panel look like? Like, will there be a standard panel to go, you know what, your T cells are exhausted and you're not going to respond to the bispecifics or whatever they are?
I think the immune system is so dynamic that we have to be thoughtful about, doing one single test and thinking we have all the answers. What my immune system is doing today is going to be different from what it's doing tomorrow. So, if I was to just take a test today, I don't think you can define it for the rest of my life, basically. And we need to be thoughtful about not putting patients in a position where we say, okay, we've defined it once and we're done and you don't get a treatment. That wouldn't be right. I don't think from an immunotherapy standpoint, that would ethically be working for anyone, for the patients, for the science, for the drugs. So let's just be very careful in how we come up with those kinds of tests and how we mandate them. But I do think, that we can start to have an understanding of what kind of therapies we can give based on what the immune system is doing at that time period, right? So for example, maybe patient who has a particular immune response that's ongoing with some exhausted T cells and some myeloid cells, get a combination of drugs that can target an exhaustion marker plus something to target myeloid cells. Whereas another person who has more naive T cells gets a different kind of drug cocktail that will help, jumpstart the naive T cell response. I mean, I think, we can come up with ways of figuring out the personalization of the therapy. I don't know that we could potentially come up with tests, and I hope we wouldn't come up with tests that say, oh, well, you don't get any therapy at all. I just don't see how that would be beneficial.
Yeah. And how about the efficacy in solid tumors versus hematologic malignancies? Do you see a big difference in immunotherapy in either of those?
Well, it depends on what you mean by immunotherapy. If you mean the checkpoint blockade unleashing your T cells, no, I don't really see a fundamental difference. But with CAR T cells, there's a huge difference because, as we were saying, as Pam was saying, B cells, for example, B cell leukemia, they go through certain steps of development, all B cells, and they have different markers on their surface that are only on B cells. They're not anywhere else. Genetically, they're not needed. They're only needed on B cells. So you could wipe out all the cells with those molecules and it doesn't hurt you. They're just, in solid tumors, it's not quite that simple because, cells are subtly different. And they all have to have ability to make energy and make DNA and make protein and all this stuff and get growth factor receptors and regulate their growth and everything. And those are shared pretty much. And you've got to be really careful when you pick one of those to attack, because it could be on normal cells somewhere in the body and you won't necessarily know it.
Yes, I think from the immune checkpoint therapy, the anti-CTLA-4 and the anti-PD-1 and PD-L1, you see that they've had predominant responses in solid tumors because they're not targeting the cancer cell, right? They're targeting the immune system, and then the immune system is targeting the cancer cell. So the immune system can select basically the mutations that are part of these solid tumors. And those are the mutations that they will recognize. And they will not recognize the wild type antigens, which are part of your normal cells. They'll only recognize the mutated antigens. So you can see that immune checkpoint therapy is allowing that to happen. And this is why solid tumors are responding well. But, some of the hematologic malignancies are, Hodgkin's lymphoma, for example, responds very well to immune checkpoint therapy. So in the setting of CAR T cells, though, you do need a specific antigen. And from that standpoint, it's why the hematologic malignancies are responding better to CAR T cells than the solid tumors.
Yeah, it's so interesting to see which immunotherapy will work the best in which kind of cancer. So I have a question too about precision medicine. I would say when I was first diagnosed, that's all you heard about was precision medicine, precision medicine. We're gonna do the genetic sequencing of your tumor. We're gonna find it all, and then we're gonna target it. But in myeloma, there are thousands of genetic mutations you might have. It's probably the same in many other cancers. So where do you see immunotherapy either being precision medicine or like replacing this idea of precision medicine?
I guess the best way to explain it is with precision medicine, the idea was there are driver mutations. The cancer biologists look at it. There's two kinds of mutations, those that are drivers, that cause the cancer, growth factor receptors or pathways that tell the cell to divide. Anything along that pathway, if you get a mutation that locks them on, it's continually telling that cell to divide. And so, but there are a lot of those kinds of pathways. And so the problem is that with time, tumors become unstable and they generate a lot of mutations. And if your tumor consists of a few million cells, which it probably does, the chances are there's a second mutation in there already that has a driver other than the one that you're targeting. And so it's not curative. You're going to kill 99.9% of the tumor, but the other one's going to come up, and then you got to deal with that. Whereas the immune system detects the products of mutations. That's what it sees, not whether it's a driver or passenger. The immune system doesn't know the difference, doesn't care. It just says there's something wrong with this cell. It's got mutations that it ought not to be here, so we're going to kill it. And and there may be 400 of them.
Yeah, that's why I like this approach.
Yeah, but I think the way to move forward is combinatorial approaches, right? Where you take advantage of both a drug that targets the tumor cells based on specific mutations, and you combine it with an immunotherapy agent. Because again, the immunotherapy agent is not necessarily targeting the tumor cell itself. Right? The drug, the immune checkpoint therapy drugs are targeting the immune system. And so if you have the two, one targeting the tumor cells to allow for some death of tumor cells, which creates the initial inflammatory response and tumor cell death, then you can say, okay, you generate this initial small immune response that then an immune checkpoint therapy agent, for example, can enhance further and take advantage of that to drive the anti-tumor response, even hopefully to a complete response.
Yeah, another way of saying that is your drugs that you go in with the idea of killing a tumor cell. You've got to kill every tumor cell with that drug, or you lose. But if you get the immune system involved, you don't have to kill every tumor cell. You just kill enough to alert the tumor cell,
The immune
cells, alert the immune cells that there are bad guys there, they're bad actors, and the immune system will take them out. Same's true of chemotherapy, targeted therapies, radiation therapy. They need to be changed to where you don't try to kill every last tumor cell. That's what causes all the side effects and all the problems that are associated with them as well, trying to kill every tumor cell, because you can't really. And if you try, you're just going to do damage. And let the immune system help you.
And now these days, people are even saying the chemotherapy that works and radiation actually is also taking advantage of the immune system, right? They're bringing in the immune system. So it was doing it without an immunotherapy drug. Now we have the ability to add an immunotherapy drug to do it even better.
I have a lot of questions about application of immunotherapy, so I'll ask those in a second. But Dr. Sharma, I wanted to ask you too about this. You mentioned this microbiome that's in your immune system. They know that's the case, that the microbiome is involved in multiple myeloma. They're not sure exactly how, you have all these factors that are happening in the bone marrow, but, and there's a lot of, patients they probably always ask you, like, what can I do with my diet? Or what can I do to change my microbiome? I'm sure you hear that a lot. Do you want to address the relationship between that tumor environment with what's happening there?
Yeah, I mean, obviously, we all know, right, that sleep, diet, stress, all of these things impact your immune response, caffeine, alcohol. So we have a general sense that things will impact your immune response. It's part of, how we're made up. The problem is that we don't have the specific mechanisms. We don't know exactly which microbiome impacts which particular T-cell response or which particular myeloid cell response. So although we have a growing knowledge base to indicate that there are these interactions, there's still a lot of work to be done to really, look at molecular mechanisms. And I think once we have molecular mechanisms, then we can make recommendations about, you have to have this particular protein in your diet because it will stimulate this particular bacterial strain, which will help with this particular T-cell response. I don't think we have that right now. And the most we can say is, we want everybody to live a healthy lifestyle. And I wish I could define that better other than to say, do what your mom always told you, eat your vegetables, get some fiber in your diet, get 8 hours of sleep, drink lots of water, get some exercise. But I don't think, we know the specifics yet.
Yeah, okay. It's a good answer, and I think we just don't know yet. Let's talk about sequencing a minute, because in your previous comment, you talked about potentially maybe using a targeted inhibitor or some kind of normal alkylating agent or something like that to reduce the tumor burden, and then hit it with an immunotherapy. Like we've seen in myeloma, they tried checkpoint inhibitors with the immunomodulators, and that was a disaster. Mm-hmm. So they're trying checkpoint inhibitors with other things in myeloma, which might be great. But then you're combining two different types of immunotherapy. So how do you proceed as scientists with that complexity? And how do we get to a point where we can go, okay, you should do this first and then this, and follow up with this? And
I think, again, for myeloma specifically, I want to discuss that myeloma really is happening a lot in the bone microenvironment, right? It's one of the areas that we have to be thoughtful about how this disease actually is within a specific niche. It's not as if it's growing in the liver or in the lungs in the same way as, other tumor types. So what do we understand about the bone microenvironment? How the immune cells get into the bones? How do they differentiate? What are the cytokines? What are the signals? Those are still question marks that we're asking, in research labs that we don't have all the answers to. And what we've done is we've taken, this works in melanoma, which is, growing in melanocytes in the skin or in lymph nodes or in soft tissues. And then we've tried to apply that knowledge into multiple myeloma. And that, I'm not saying there's anything wrong. We had to take our best educated guess and move forward, but knowing that it didn't work the way we expected to really forces us to go back and say, what is different? What is this microenvironment dictating to us that may require a different treatment strategy? And, we had a similar study because we saw patients with prostate cancer who had bone metastases. These patients were not responding well to immune checkpoint therapy because they had bone metastases, and that's where predominantly prostate cancer goes to, is the bones. So we had to set up really the research question to ask about what's happening in the bone microenvironment there. And when we did, we saw very different signaling from cytokines that skewed the T-cell response in a different direction in the bone metastases versus melanoma, for example. And that really gave us an inkling of how we may want to tackle treatment of these patients, and we're developing a new clinical trial based on that data. But I think the same applies here. It's really getting back to doing the basic research and getting the biopsy samples from the myeloma patients themselves to say, what was it that we did target and what was not targeted appropriately, or how did the immune system not respond to immune checkpoint therapy in myeloma? When you compare myeloma responses to melanoma, how is melanoma different from myeloma? How is myeloma different from melanoma? What can we learn from those kinds of studies that then tell us what needs to be targeted in multiple myeloma? So I think we have a long way to go, but and there are really excellent labs asking these questions. We just have to make sure that we keep funding those kinds of research studies.
Yeah. Similarly in AML's made a lot of progress since 2017, and you see a lot of new immunotherapies coming into the AML space. Do you have any comments about AML in general in immunotherapy?
Yeah, I think in AML too, we're starting to see, we published some studies with Naval Dover and the leukemia group here at MD Anderson in AML. So we've seen some data that, the immune checkpoint agents may be able to have in combination therapies with some of the other AML therapies, that we do see some responses now. We're beginning to explore that in larger cohorts of patients. There are also data looking at some of the bispecifics and some of the newer therapies as well in AML that looks promising.
Mm-hmm. Your research has been so much on T-cell work, and I know NK cell work is coming up, and I don't know a lot about NK cell research. Do you have a feel for T cell therapy versus NK cell therapy, are they to be used in different instances, or it's just that starting? It seems like it's a little further behind T cell
Yeah.
work. Or,
I think, again, a lot of this is a little bit further behind because, again, we know so much about regulation of a T cell response. We know the basics. We know the T cells need signal 1, which is the MHC plus antigen. It needs signal 2, which is CD28 and B7. And those things together will drive CTLA4, which needs to be blocked if you're going to continue that response. There's so many more details about the molecular mechanisms of how T cell responses are proceed. Versus NK cells at this point. But really, we're getting more and more data every day, and that's going to help us figure out how to target NK cells. I do think at some point, though, T cells are truly the soldiers of the immune response. So all these other cell types, yes, we need them, and they have to work in concert with T cells. So we have to figure out how we get all of these cells to, when we give a treatment strategy, whether it's a combination therapy or other therapies, we need to know how those combinations are impacting T cells as well. So maybe they are directed to an NK cell, but hopefully they're bringing in those soldier T cells when we give them so that they can do the job.
vice versa, like some monoclonal antibodies in the myeloma space, like they reduce NK cell function, and like, is that good or bad? Like, I don't know.
Well, like T cells, there are two sides, right? So NK cells have cells that help the immune response, and they may have cells that suppress an immune response. And T cells have T cells that are good T cells. They're the effector T cells that will help tumor rejection, but they're certainly regulatory T cells that are the ones that will suppress the immune response, right? So there are two sides. So when we're talking T cells, we're not always talking the effector T cells. We have to be thoughtful about how we get rid of the regulatory T cells. So it might be the same in NK cells as we go forward.
The most interesting data, I think, doesn't support really that NK cells necessarily physiologically do much to tumors themselves, but they recruit a lot of T cells to come in and do the job.
Oh, got it.
By the T cell response. Now, whether putting in a ton of NK cells will do that, we'll see. There's obviously some of our colleagues here are doing that.
Okay, so interesting. A patient asked, there seem to be a lot of targeted therapies in the myeloma space, I'm sure other cancers, like BCMA, CD138, et cetera. But how can you identify which of these is right for a particular patient, and how do you think that will happen over time?
I think these therapies, again, as they're out there, your physician will be the one to tell you, right? There are all these tests that have to be done. So if you're getting a BCMA CAR specific T cell, for example, you need to have a BCMA positive tumor, and they will take a biopsy and they will test that, and they'll make sure for those things, and again, multiple myeloma is one of those settings where the antigen is expressed. So a lot of times maybe we don't even need to do some of the tests because we know that from lots of other studies that have been conducted. But there are ways in which your physician will continue to monitor you and your tumor as it evolves to pick the right therapy. And so I think that's going to be what it is. It's not going to be a one size fits all.
Mm-hmm. And I asked this question before we got on, but looking at, in your documentary, you talked about how you were kind of borrowing some of the research a little bit from work you'd done in autoimmune disorders and bringing it to oncology. How do you do that to look cross disease at the immune system that is so complicated and bring those types of effective things? Like, we even did a study with myeloma and psoriasis to see if there was a correlation, because that was like the highest autoimmune disorder in myeloma patients in our database. And so we went to see, like, okay, will an IL-17 inhibitor kind of work in myeloma? And for me, it did not, but because I have both psoriasis and myeloma. But how do we do that more effectively?
I think it's about collaborations, right? If there's anything we learned from the pandemic, for example, is how to bring a broad and diverse group of experts to the table so that they can all help. If you think about the pandemic, we brought in vaccine specialists, we brought in virus specialists, we brought in people who specialized in, myocardial or heart issues because people were worried about possible side effects. We brought in people who were respiratory or pulmonary physicians and respiratory experts to talk about how the lungs change. So we brought a different sets of experts to the table to help us understand the best ways in which to target the virus. Initially, we didn't have a vaccine. We were just trying to, get people through symptoms, right? Help them to breathe better, help them to get rid of the inflammation, and eventually the vaccine could be developed and we move forward. So I think it's the same thing here, right? Cancer is a broad, diverse group of diseases. Actually, we use the word cancer, but it encompasses so many different principles, everything from immunology to, as you mentioned, autoimmune and cancer, within that immunology space have to speak to each other. The nervous system, makes up part of this. There are lots of different areas where if we can bring the experts to the table and have that conversation, we can learn from them. Jim and I are not autoimmune experts at all, but we learn so much from the autoimmune experts when they share their data about what's happening and what's going on in their research laboratories. Then we can have ideas, and our students can have ideas about things to test in our laboratories on the cancer side. So it's how do we convene these groups at scientific meetings and not make a scientific meeting, for example, just about cancer? Like, how do you make it about broad, diverse areas so that students are really learning from each other across these different groups? I think that's very important to do.
Yeah, that's so interesting. And Dr. Allison, when you look at when I look at disruptive innovation, it usually comes from an individual. And so that's where collaborations versus individual curiosity and somebody who just wants to push something forward. How do you balance that idea of collaboration with just the driver? Because that's how a lot of disruptive innovation just happens. You have this person like you that's just doing it.
Yeah, I mean, I think you stated it. I think that if you have important issues and you need to discover, a committee's never going to find them. They're
Right,
not.
right. It's like the
cancel each other out.
project in school, right?
On the other hand, if you say, this is the way, we got this course idea, and if you do it this way, it's going to work. And you say, get a whole bunch of people together and refine it and refine it and refine it. Then there's a time for that, there's a time for it, but it's not discovery science. It's enabling science. And that's kind of where I draw the line. You discover or you enable. Mm-hmm. And they're very different skill sets.
It's a different personality too, I think, sometimes. Because sometimes people who are more risk averse and it's tough because you get to a certain point in your career and then it's tough to have some things that don't work or whatnot, and you've tried so hard and made it, and I don't know, it's to me it's not a system that rewards this type of innovation. We need to change that somehow.
And there's so much talk about team science now, and it's valuable, but if you stress that too much, I'm worried that you're going to lose the people that'll say, hey, look at this, look over here, you're looking at the wrong place, look over here.
Yeah, totally. So I want to talk about becoming refractory to immunotherapies. What makes that happen? Dr. Allison, maybe you want to start, and then Dr. Sharma, I know you've done some research on this.
Yeah. Oh, in a lot of ways. You can lose the target. Now, T cells, if the target the T cells are attacking is not essential to the survival of the tumor cell, it could just lose it, and then the T cells won't attack anymore. You can cover that to some extent by having a whole lot more different targets on one cell where they're not likely to be able to lose all of them. But then you can also have things that have, myeloid cells, so we keep coming back to that can come in and keep T cells from functioning right. And even if you have them, they may not function properly because you've got other things that are making the myeloid cells dominant. That seems to be something that happens in pancreatic and glioblastoma. And so that you need to get, figure out how to, get them out of the way. Or the regulatory T cells that Pam talked about, they can come in and turn off the good guys, and we're learning ways to get rid of them, but it's just, there's a push and it's like whack-a-mole in a way, immune system.
Right, totally. Well,
It's gonna respond, and we just have to be smarter.
It's a yin and a yang, right? The yin, you push the immune system in one direction, it will try to control itself. It has to, because otherwise you'll end up with autoimmune disease if you allow it to continue down the path. So it must have intrinsic mechanisms to control itself. So what are those intrinsic mechanisms? How do we understand when they come into play? At which time point within an immune response? Is it day 1? Is it day 5? Is it day 12? Do you have a different regulatory response that tries to control the immune system, at which day and time and which microenvironment? Is it within the bones? Is it different versus within the brain versus within the liver? So, it's a complex system made up of many cell types that require signaling mechanisms. And we have to know when those signals are at play in order to really target each of them at the appropriate time and within the appropriate microenvironment.
yeah, you need more information. It sounds like a lot more information.
Yes.
If we're gonna cure these diseases, you need to know what's happening at every step
Yes.
and how it's being affected. Wow. Okay, I have 2 more questions because we're running short on time. The first is about CRISPR. Where do you see gene editing and CRISPR going? And Dr. Allison, maybe you want to start.
Well, I think there are ways of using it therapeutically, but I think right now it's a discovery tool, because you could take out very quickly, very, there've been ways of disabling genes around for a long time, but not nearly as fast and as neat, crisply, I guess I say it's crisp, that can give you discovery tools. You see, take out something, do you gain something or do you lose something? And it can help you find new targets that way. I think for genetic diseases, you could, eliminate bad genes, maybe replace them, with repaired genes. That can certainly be done. But right now, I think it's primarily a discovery tool.
I mean, you can see that it's even coming over into where immunotherapy is, right? Because with the CAR T-cell work that Carl June's lab's been doing, they were able to CRISPR out PD-1, for example, in those T cells, so that those T cells are no longer under the inhibitory signals that a PD-1 PD-L1 interaction may allow. So they were testing out those CAR T cells, for example, to see if you can make better CAR T cells. So I think they're still testing that concept, but clearly that would not have been possible without CRISPR. So these fields will start to interact, and we will see more and more data coming out as people are testing new concepts.
It's more on the basic science side still. Yeah. Okay, to see what's going on or what impacts what when you do what, Yeah. Okay, the last question, and Dr. Sharma, let's start with you, and then Dr. Allison, you can have the final word. What in terms of new immunotherapy research excites you the most?
I'm very excited to see some of the new combinations that are being tested. I'm very excited to see that we're actually thinking of how to target myeloid cell differentiation, for example. I'm, super excited to think about triplet combinations, not just doublets. And I'm truly excited about the concept that, we are looking for patients to participate in the research studies with us, so that we're doing 10 or 20 patient small trials where we're not doing hundreds of patients, but just small numbers where we can get access to the samples and really ask the question about how these drugs, these combinations or triplets are affecting the tumor microenvironment. How is it affecting the immune response at different time periods, day 1 versus day 10, so that we can come up with, new ideas and new concepts for the next round of combination therapies with more effective drugs, hopefully.
Okay, great answer. Dr. Allison?
I would say largely what Pam just said. I mean, she was one of the ones that pioneered the idea of comparing the immunological state pre-treatment versus post-treatment to learn how the immune system responded, to tell you what you need to do next. And so that's incredibly powerful. But I think that we've entered into this period now, and I think it's partially the fault of people who just, follow bandwagons and stuff, where it's all immunotherapy, and forgetting about something that I've been fascinated. I mean, all of the immune responses start with tumor cell death, a little bit. You don't need to kill every tumor, you just need some, cause inflammation, and the immune system can take off. And yet we're studying all these checkpoints that are regulating them, which is incredibly important. But I think that one of the things is that if we know exactly when the immune system starts working, which is when tumor cells are dying in one way, at least the early steps, then we can design much more precise ways of shaping the way that immune response is going to come out.
And that means, yeah, like using what you were saying earlier, using maybe some chemotherapy, maybe some immunotherapy, maybe some and then figuring out which sequence to put that in.
Yeah, just something that, if you've got a kind of cancer and you've got a drug that works pretty well, then find out next, how does it impact the immune system? Does that drug kill T cells? If it does, that doesn't mean you can't use it. It just means you got to think about the timing and, and the dosing, et cetera, but we need to be more clever about putting multiple modalities. People say, "Oh, well, is chemotherapy a thing of the past?" I don't think so. I think that as a monotherapy by itself, it is. It's going to have to be combined with immunotherapy, but I think all these things have a place.
Well, I had a previous question that I didn't ask already about bringing immunotherapy up front line, and that kind of goes to that question, like, do you do that? Is that the right move? Is it not? I don't know.
We don't, I mean, we think, again, all of these are going to be dependent on which cancer type we're talking about, right? Which microenvironment are we talking about? These need to be tested in clinical trials in patients for us to really have an understanding. And maybe in some setting like lung cancer, chemotherapy comes first. In other settings, it comes later. But we need to really do the work and get those answers before we have large numbers of patients, exposed to these treatments.
Yeah. In melanoma, for example, there's been a couple of fairly big phase 3 trials that have been followed for time. And one of them was just reported on a few weeks ago, shows that 58% of the patients that get a combination of anti-CTLA-4 and anti-PD-1 are alive 6.5 years after treatment, whereas historically it was less than 3%. And most people died within 7 months. So,
Yeah. Well, I said I wasn't going to cry, but maybe I will for a second.
We got a long way to go in a lot of the cancer and melanoma as well. But.
Yeah. As I just want to thank you both for joining. I'm headed to my CAR T therapy tomorrow, and I want to thank you for making good things popular in the world.
Wishing you all the best of
like
success.
Immunotherapy. So just thank you so much for everything you've done in this space, for the continued research that you continue to do. Patients everywhere are so grateful.
Thank you. Thank you all for having us today.
Yeah, that's why we do it.
Thank you.
Great. Thank you guys so much. I learned quite a bit, and I really appreciated the information and the passion that was shared today. I will close this meeting with just a couple of announcements, and then we will be done for today. So thank you again for your participation today to all of our attendees. You may be interested in myeloma crowd community events that you have coming up. We're low on time, so I won't get into too much detail, but tomorrow at 1 p.m. Eastern, you can join the Connect with the Myeloma Crowd chapter to learn more about HealthTree Cure Hub and different features you won't want to miss, such as personalized treatment options, personalized clinical trials, how to find your myeloma twin, and how to connect with others through community forums. Wednesday at 1 p.m. Mountain is our Mountain West Myeloma chapter. Huntsman's newest multiple myeloma specialist, Dr. Manny Mohudin, is going to summarize some of the most exciting and thought-provoking multiple myeloma research shared this year at IMW, the International Myeloma Workshop. Thursday at 1 p.m. Eastern is the newly diagnosed myeloma patients chapter. We will provide a way to connect with other newly diagnosed myeloma patients, as well as learn how to properly advocate for yourself if you're in this position in the healthcare setting. The link to sign up for any of those events and even more events I have not mentioned today is found at the bottom of this slide and will be sent out in our follow-up email within 48 hours of today's event's conclusion. Another thank you to our sponsors, Bristol Myers Squibb, Takeda Oncology, Karyopharm Therapeutics, Adaptive Biotechnologies, Oncopeptides, Janssen Oncology, AbbVie, Sanofi, Amgen Oncology, and Genentech. Thank you again to Dr. Allison, Dr. Sharma, and Jenny for helping moderate the session today. We appreciate you all and hope that you have a great rest of your day. Thank you.